biomedical waste management system. As a microbiology student, I want to observe how infectious waste is handled and treated on industrial scale. Another objective was to learn the operation of specialized equipment such as autoclave, shredder, incinerator, and effluent treatment plant. I also want to understand environmental regulation compliance requirement related to biomedical waste management. A major goal was to connect microbiological concepts such as sterilization, disinfection, contamination control, and pathogen restriction, which real industrial practices. Now, work assigned during my internship. During my internship, I was assigned to observe various biomedical waste treatment processes. I observed the collection and segregation of biomedical waste according to color coded categories. There are three types of color categories. First one is yellow bag, red bag, and white bag. I monitored autoclave operation, including sterilization cycles and parameter verification. I studied the functioning of shredder used for size reduction of treated waste, which is efficient by red bag. I observed incinerator operations, including waste feeding procedures, temperature monitoring, and emission control system. I also visited the effluent treatment plant and studied wastewater treatment processes. In addition, I reviewed documentation practices which is involved in autoclave and incineration, safety procedures, and PPE compliance. Now, about autoclave operation. In autoclave operation, the work performed is one of the most important sections I studied. An autoclave is used for sterilization, sterilizing infectious biomedical waste using high pressure saturated steam. During my observation, I learned about waste loading procedures, cycle operation, unloading procedure, and parameter monitoring. I observed critical parameters such as temperature, pressure, and holding time. These three parameters are major role of autoclave. Typically, sterilization is achieved at temperature above 121 degrees Celsius under pressure for a specific duration. It may be 15 psi. I also learned about biological indicator and chemical indicator used to verify sterilization effectiveness. Now, from a microbiology perspective, autoclaving is highly significant because it destroys bacteria, virus, fungi, and spores before further processing. Because the main role is to kill bacteria, virus, fungi are those present on the biomedical waste are killed by autoclave operation. Now, shredder operations. In shredder operation, after sterilization, first we have learned about autoclave. Now, after autoclave step, we have to next perform shredder operation. After sterilization, the treated waste is transferred to shredder. The primary purpose of shredding is to reduce the size of waste material and prevent unauthorized reuse of medical items. I observed the feeding machine, safety precaution, and operational procedure of shredder. The machine converts larger waste material into smaller fragments, making them unsuitable for reuse and easier for final disposal. Now, in incinerator operations, the incinerator is one of the most critical components of biomedical waste treatment. It is primarily used for yellow category waste that cannot be recycled or sterilized effectively. Now, in yellow category bag, it involves blood contaminated parts, which will come from hospitals, clinics, or blood banks, etc. I observed waste feeding operations, combustion process, and temperature monitoring. The incinerator consists of primary chamber and secondary chamber. The primary chamber's temperature is required for 850 to 900 degrees Celsius, and the secondary chamber is at 1050 degrees Celsius, um, 1050 degrees Celsius heat. The incinerator consists, okay, the primary chamber initiates combustion. The role of must primary chamber is combustion, and the role of secondary chamber is ensure destruction of harmful gases. Harmful gases produced in primary chamber, those are killed by in secondary chamber via producing 150 degrees 1050 degrees Celsius temperature. High temperatures destroy infectious pathogens and significantly reduce waste volume. I also studied air pollution control system, which minimize emissions before gases are released into the atmosphere. Now, in pollution control system, uh, in that incineration involves quencher, venturi, mist eliminator, and on. Now, in effluent treatment plant. The effluent treatment plant is responsible for treating wastewater generated during waste treatment operation. I observed different treatment stages including primary collection, aeration, settling, and chemical treatment. Now, in aeration promotes microbial activity that helps to degrade organic pollutants. Settling tanks remove suspended solids from the wastewater. Now, in chemical treatment, in the method of chemical treatment is used to neutralize contaminants and improve water quality. Now, the question is, what chemical should we give to neutralize the chemicals? In my in those areas, I have learned about alum and lime are the chemicals which are used to neutralize to neutralize the effluent. And I learned that wastewater must meet prescribed standards before discharge into the environment. The ETP plays a significant role in preventing water pollution and ensuring environmental compliance. Now, field observation in area. The field observation was that proper segregation at the source is foundation of effective biomedical waste management. Incorrect, somehow incorrect segregation can compromise the entire treatment process. I also observed that consistent use of PPE kits significantly reduce occupational health risk for workers and employees. I also observed that mm, okay, regularly maintenance of equipment improves operational efficiency and minimizes breakdown of microorganisms. Now, another important observation was the critical role of documentation in maintaining regulatory compliance and operational transparency, which was given by GPCB rules. Finally, I observed that proper treatment process effectively reduce infection risk and environmental contamination. Now, during overall internship, I have gained technical knowledge. Now, here are the technical knowledge. During the internship, I developed a better understanding of biomedical waste management tools 2016. I learned the significance of color coding and waste categorized system. I understood the working principle of primary and secondary chamber in incinerator. I learned about APC system and their role in reducing emission. I also studied multi-stage wastewater treatment process in ETP. Furthermore, I observed the importance of record keeping, monitoring instruments, and operational documents in maintaining regulatory compliance. Now, in key learnings, major key learnings I have get from during internship are it gave me practical exposure to biomedical waste treatment system. I learned the importance of workplace safety and PP usage and hazard prevention. And the internship helped me to understand how microbiology is applied in waste management industry. I realized that the importance of sterilization and disinfection in controlling infection areas. Conclusion: the internship strengthened my knowledge of microbiology while introducing me to industrial application and environmental compliance practice. I would like to express my Sincere gratitude to GPCB Navsari, the management, supervisor, and my faculty member for providing me with this excellent learning opportunity. Thank you.
Okay, fine, Mr. Vidyasagar. So, you have referred biomedical waste management rule 2016.
Yes, ma'am.
Okay. So, what is the color coding in the biomedical waste management?
Color coding means there are three types of color codings, which is red bag color
Mm,
coding, yellow bag color coding, and
mm.
white bag color coding.
There's also
This
blue bag.
blue bag, but
Yes,
those in
blue
GPCB
container, not
Navsari,
bag.
yes, ma'am.
Yeah. So, refer it again. You will find all the color coding category. Okay.
Yes, ma'am.
And color code wise segregation of waste. Okay.
Ma'am. Yes, ma'am.
Now, tell me what is the function of autoclave,
The
which you have
autoclave
just
functions
shown in this
mainly to
presentation.
sterilize mainly used to sterilize the biomedical waste.
Sorry, repeat again. Your voice is tracking.
Autoclave is mainly the main function of autoclave is to sterilize materials, biomedical waste.
What is sterilization?
Sterilization is free is the process the material which we have to make free of microorganisms or pathogens.
Hmm. Have you seen the process of sterilization?
Yes, ma'am.
So, what chemical they are using to sterilize the medical waste?
No chemicals, biological indicators.
Yeah. So, can you name them?
Stereothermophiles, geostereothermophiles.
Okay. Okay, fine. And
Yes, ma'am.
what records or documents we need to maintain for the compliance of biomedical waste management rule?
We have to maintain the rules according given by GPCB.
Yeah, but what are those records or documents?
Those records are different to different types of um, instruments, such as uh, there is different rules for incinerator, different rules for autoclave, and different for ATP.
Yeah, you are quite correct. But I'm asking about the records or documents. It is not only for instrument; it is for waste also, biomedical waste, right?
Yes, ma'am. I don't know about this.
Okay, fine. Okay. Any other question from Dhruvi?
Yes. What is the difference between sterilization and disinfection?
Disinfection is carried out on the surface, and sterilization is used for the free of microorganism for any material.
What are the commonly found pathogens in the biomedical waste?
What?
What are the commonly found pathogens in the biomedical waste?
Uh, I don't know.
Okay. Do you know what is the temperature of autoclave
Yes, 121 degrees Celsius,
and pressure?
15 psi.
Okay. If the sample is liquid, then if the sample is blood, so it goes to the which color of bag?
It goes to yellow color bag.
And what we do with that yellow color bag waste?
We should do incineration.
What is the temperature of incineration?
In primary chamber 850 degrees Celsius, in secondary chamber 1050 degrees Celsius.
Okay, done from my side.
Okay. Mr. Vidyashagar, you have not submitted your final report.
What, ma'am?
Your final report is yet to be submitted.
I will send you,
You have
ma'am.
only submitted PPT presentation.
Yes, ma'am. I will send
So
you.
you told me that you are placed at some industry in
Yes,
Navsari, right?
yes, ma'am. Yes, ma'am.
So what about that industry?
That was DPCB. I mean, that industry, Samved Biomedical Waste Management Industry, that was also authorized by GPCB.
So you have not mentioned here about your internship place.
Yes, ma'am. Because my internship came from GPCB in Navsari night. That's why I'm not mentioned.
But you could have taken permission from that health biomedical waste management facility
Yes, ma'am.
and
Yes,
upload
ma'am.
your
Of course,
PPT there.
I have taken permission from that industry HR.
Yeah. So you could be able to showing some photographs of that facility here.
Ma'am, they didn't give me photograph because it is prohibited. They said photographs are prohibited. That's why I'm not taken.
Okay, no issue. Um, is there any website of that facility?
What, ma'am? Hello.
Hello.
Ma'am.
Is there any website of that facility where
Uh,
you have
yes,
pursued your
ma'am.
internship?
Yes, ma'am. Website Samved Biomedical Waste.
Yes. Okay. Okay. Fine.
Should I share in your
Okay.
WhatsApp?
So let us move ahead. Yeah. Please share me its detail on WhatsApp. Okay.
Okay, ma'am. I will send you.
Our next intern is Mr. Dhruvil Upadhyay. Dhruvil, please start your presentation. Share your screen.
Hello.
Yes, you are audible.
Amen. So, good afternoon, everyone. My name is Dhruv Upadhyay. Today, I am presenting my internship report on presentation on e-waste management, which is completed at GPCB Gandhinagar. This presentation covers e-waste generation, aim management, and practices and legal framework and suggestion and sustainable e-waste handling. Uh,
Yeah, continue.
Amen. So e-waste is a refers to discarded electrical and electronic devices such as mobile phones, computers, printers, televisions, batteries, etc. Due to rapid technology advancement, the generation of e-waste increases every year. Uh, a improper disposal can harmful for human health and the environment. Therefore, proper e-waste management is essential for a cleaner and safer safer future. So, e-waste management is this slide explains three important aspects. First is the e-waste management recycling process, and E-waste management rules is 2022 promote collection, recycling, and safe disposal of e-waste throughout extended producer responsibility. Second one is Gujarat generates approximately 75 to 80,000 uh, metric tons of e-waste annually, with major contribution for Ahmedabad, Gandhinagar, Surat, Vadodara, etc. Third one is a CPCB and role of CPCB and GPCB. So CPCB is develops national guidelines, and while GPCB is implementation and monitors e-waste management activities within Gujarat. Uh, Gujarat. So our next slide is a legal framework and SOPs. So, e-waste management in India is governed by Environment Protection Act 1986 and the e-waste management rules 2022. These regulations ensure proper collection, recycling, and disposal of electronic waste. Standard operating procedures include collection, uh, uh, separate collection, storage, dismantling, recycling, material recovery, hazardous waste, and record uh, maintaining proper records of e-waste. So next slide is resource recovery and benefits. So e-waste contains valuable materials such as copper, aluminum, gold, plastic, silver, glass, etc. So throughout recycling, this material can be recovered and reused. Proper e-waste management reduces pollution, protects human health, and conserves natural resources, save energy, and support the circular economy. Suggestion and improvements. To improve e-waste management, collection system should be uh, strength and more awareness programs should be conducted. Extended procedure responsibility should be implemented effectively. Citizens should use authorized collection centers and dispose of e-waste. E-waste responsibly. Better coordination between the formal and informal sector can further improve recycling efficiency. So, uh, proper e-waste management is essential for environment protection and sustainable development throughout effective regulation, public awareness, and responsible recycling practices. So we can reduce pollution and recover valuable resources. Thank you for everyone for attention.
Okay, Dhruvil. Very good presentation slides made from ChatGPT. We must appreciate that. તમે મને પહેલા જે પ્રેઝન્ટેશન સબમિટ કર્યું હતું એ તો કંઈક અલગ હતું.
હા મેમ એ 22 સ્લાઇડનું હતું ને 23 સ્લાઇડનું એટલે મેં જરાક શોર્ટ કરીને કર્યું હતું તમને તમે મને કીધું હતું ને તો
બરાબર. ઓકે. તો ઈ વેસ્ટ મેનેજમેન્ટનો આ તમે જે ટોપિક કવર કર્યો છે બરાબર. તો ગુજરાતમાં કેટલા રજિસ્ટર્ડ ઈ વેસ્ટ રિસાયકલર્સ છે? ખ્યાલ છે?
હા મેમ હેલો
હા
હા મેમ આઈ થિંક
મને સર્ચ ના કરશો, મને
હા, હા.
ખ્યાલ હોય તો હા પાડો, ના ખ્યાલ હોય તો ના કહો. ખબર ના હોય એ કોઈ ગુનો નથી, તમે પછી લેટર ઓન બી
ના.
સ્ટડી કરી શકશો બરાબર.
ના મેમ આઈ થિંક 300 અરાઉન્ડ છે. મને એક્ઝેક્ટ ખબર નથી, મેં વાંચ્યું હતું બટ એક્ઝેક્ટ નથી ખબર. 300 ની આસપાસ છે એટલી ખબર છે.
વેરી ગુડ.
હમ.
હવે ઈ વેસ્ટ મેનેજમેન્ટ જે રૂલ્સ છે બરાબર, તમે જે કવર કર્યું પ્રેઝન્ટેશનમાં બરાબર, હવે એના ઇમ્પ્લિમેન્ટેશન માટે કઈ વસ્તુ છે જે મેજર હર્ડલ છે કે જેના લીધે પ્રોપર ઇમ્પ્લિમેન્ટ નહીં થઈ શકતો?
મેમ મેજર હર્ડલ ઇઝ આઈ થિંક લેક ઓફ પબ્લિક અવેરનેસ છે એક તો. પછી ઇનફોર્મલ સેક્ટર બહુ છે આપણા અહીંયા જે કહેવાતું હોય ઇનફોર્મલ સેક્ટર જે ડાયરેક્ટ એ કરતા હોય જે. પછી મોનિટરિંગ થોડુંક વીક પડતું હોય. પછી અને કલેક્શન આઈ થિંક કલેક્શન એન્ડ રિસાયકલિંગ થોડું ઓછું છે, એના માટે અવેરનેસ થોડી વધારવી જોઈએ.
બરાબર. ઓકે, બીજું કઈ?
ના મેમ બીજું તો કઈ અત્યારે નથી યાદ આવતું.
તમે કોઈ ઈ વેસ્ટ રિસાયકલિંગ ફેસિલિટીની વિઝિટ કરેલી છે?
ના મેમ, ડ્યુરિંગ ઇન્ટર્નશિપ તો નથી કરેલી મેમ.
ઓકે, સોરી, તમે કઈ કોલેજમાંથી શું કરી રહ્યા છો?
સ્વર્ણિમ સ્ટાર્ટઅપ એન્ડ ઇનોવેશન કોલેજ છે. બીટેક પરસ્યુ કમ્પ્લીટેડ છે, કેમિકલ ઇન કેમિકલ એન્જિનિયરિંગ.
ઓકે, તો કેમિકલ એન્જિનિયર થઈને કેમ ઈ વેસ્ટ જેવો ટોપિક સિલેક્ટ કર્યો?
મેમ આજની તારીખમાં ઈ વેસ્ટ એક મેજર પ્રોબ્લેમ છે. એટલે એ આજે અત્યારે જ્યાં જોવો ત્યાં ઈ વેસ્ટ નું વધારે થોડું એ હોય છે. આપણે યુઝ કરતા હોય ઓફિસમાં હોઈએ, ઘરે હોઈએ કે કોઈ બી આઈટી કંપનીઝ હોય કે એમાં ઈ વેસ્ટ નો ઉપયોગ બહુ જ હોય છે. એની રિસાયકલિંગ બરોબર થતી હોય ના થતી હોય એના માટે થોડું નોલેજ જોતું હતું.
ઓકે, અને તમે જીપીસીબી માંથી આ ઇન્ટર્નશિપ કરી તો જીપીસીબી ઓર એસપીસીબી નો શું રોલ હોય છે ઈ વેસ્ટ ઇમ્પ્લિમેન્ટેશન કરવા માટે, રૂલ્સના ઇમ્પ્લિમેન્ટેશન માટે?
મેમ સીપીસીબી છે એ રૂલ્સ ગાઈડલાઇન્સ બનાવે છે અને જીપીસીબી છે એ ઇમ્પ્લિમેન્ટેશન કરે છે. રૂલ્સનું રેગ્યુલેશન જે કરે છે કે મોનિટરિંગ કરવાનું છે કે પછી મોનિટરિંગ છે, પછી જે લીગલાઇઝેશન પેલું કરવાનું હોય છે, પછી શું કહે રિસાયકલર્સ જે હોય એમના ઇન્સ્પેક્શન કરવાના હોય છે. પછી આઈ થિંક અને અવેરનેસ ઊભી કરવાની હોય છે જો ઈ વેસ્ટ પ્રમાણે, ઈ વેસ્ટ ના એના. બસ મેમ.
ઓકે, ફાઇન. હવે મારો લાસ્ટ ક્વેશ્ચન છે, ઈપીઆર વિશે જાણો છો? વોટ ઇઝ ઈપીઆર?
હા મેમ, એક્સટેન્ડેડ પ્રોડ્યુસર રિસ્પોન્સિબિલિટી.
Okay. Okay. Dhruvi, from your side?
Dhruvil, are there any rules classified for imported and second-hand e-waste?
મેમ એ ધ્યાનમાં નથી મારું. હા.
Can you explain the basic steps of dismantling, shredding, and mechanical separation of e-waste? As I have seen your report and you have mentioned about all these processes, so I want you to discuss all these processes.
હા મેમ, કઈ પ્રોસેસ કીધી મેમ તમે?
Dismantling, shredding, and mechanical separation of e-waste.
ડિસમેન્ટલિંગ. મેમ ડિસમેન્ટલિંગમાં એવું હોય છે કે ડિસમેન્ટલિંગમાં સેપરેટ કરવાનું હોય છે જે હેઝાર્ડસ પાર્ટ છે, જે પેલા ઈ વેસ્ટના જે હેઝાર્ડસ પાર્ટ્સ છે એનું ડિસમેન્ટલિંગ કરવામાં આવે છે. અને પછી ડિસમેન્ટલિંગની અંદર બી બે પ્રોસેસ હોય છે. મેગ્નેટિક પ્રોસેસ હોય છે અને બીજા નંબર પર કેમિકલ પ્રોસેસ હોય છે. એ બંને પ્રોસેસમાં કરીને પછી આ નેક્સ્ટ જવાનું હોય છે. તો
No,
એ
what
જ
are
છે.
the steps? I want to know what are the steps.
ઈ વેસ્ટ રિસાયકલિંગ પ્રોસેસ કોને મેમ?
Dismantling, shredding, and mechanical separation. I want to know basic steps of this.
મેમ ખબર છે મને ધ્યાન નથી મેમ. મેં લખ્યું હશે આઈ થિંક પણ.
You don't know?
મેમ મેન્યુઅલ સેપરેટ ડિસમેન્ટલિંગ થાય છે, પછી શેડિંગમાં જાય છે, અને શેડિંગમાં જઈને પછી મટીરિયલ સેપરેશન થાય છે.
But એ shredding કઈ રીતે થાય છે?
શેડિંગમાં ઝીણા ઝીણા પાર્ટ્સ કરવામાં આવે, પેલા એના મટીરિયલ્સના જે નાના નાના પાર્ટિક્યુલમાં પાર્ટ્સ કરવામાં આવે, પછી એનું મિકેનિકલ થાય, મટીરિયલ સેપરેશન થઈ જાય.
પણ એમાં
જેને
શું
મટીરિયલ
યુઝ થાય, કયો ઇન્સ્ટ્રુમેન્ટ યુઝ થાય, કયું કેમિકલ યુઝ થાય, એવું કશું જ યાદ નથી.
ના મેમ એ યાદ નથી મને. પેલા મટીરિયલ સેપરેશનમાં યાદ છે કે મેગ્નેટિક ટેકનોલોજીસમાં મેગ્નેટિક સેપરેશન યુઝ થાય છે, એર ક્લાસિફિકેશન યુઝ થાય છે, અને ડેન્સિટી સેપરેશન થાય છે, એ ખ્યાલ છે.
What kind of hazardous substance you will found in the e-waste?
બેટરીઝ, કેપેસિટર્સ, મર્ક્યુરી, લીડ, કેલ્ડિયમ, etc.
What is PRO?
PRO.
Yes.
I don't know ma'am.
પ્રોડ્યુસર રિસ્પોન્સિબિલિટી ઓર્ગેનાઇઝેશન કેવું કંઈ સાંભળ્યું છે?
હા મેમ.
Do you know?
કન્ટિન્યુ.
Do you know e-waste amendment? When this rule amend?
હા મેમ, ઈ વેસ્ટ અમેન્ડમેન્ટ રૂલ 2023માં થયો હતો, અને 24માં પણ એનું ઇમ્પ્લિમેન્ટેશન એક્સ્ટ્રા પેલું એ થયું હતું. હેલો.
Okay, I think you need to learn more about your topic, because you have mentioned everything in the report, but you have not able to answer it. So I must guess that report must be from ChatGPT, or I don't know. So you must have some knowledge for this.
ઓકે ઓકે.
Okay, Disha ma'am, it's end from my side.
ઓકે ઓકે ધ્રુવીલ, મહેનત કરજો, બરાબર? વાંચવાનું રાખો. ઓકે.
ઓકે મેમ.
મેં
ઓકે
રૂલ્સનું બધું જ રિપોર્ટમાં, ઇવન પ્રેઝન્ટેશનમાં બધું જ ઇન્ક્લુડ કર્યું છે, બટ તમને
હા
આન્સર
મેમ.
ખ્યાલ નથી, તો તમારે રીડિંગની વધારે જરૂર છે, બરાબર?
હા હા મેમ.
ઓકે.
થેન્ક યુ મેમ.
નેક્સ્ટ કોઈ છે? કાવ્યા રાવલ ને આશ્વી કરીને, એ લોકોને પણ ઇન્ટર્નશિપ કમ્પ્લીટ થઈ રહી છે તો.
યસ મેમ.
હા, કાવ્યા તમે ચાલુ કરો છો પ્રેઝન્ટેશન?
હા, અમે બંને સાથે જ આપવાના છીએ પ્રેઝન્ટેશન.
ઓકે, સ્ટાર્ટ યોર પ્રેઝન્ટેશન.
ઓકે.
કાવ્યા, શેર યોર સ્ક્રીન.
Yeah, ma'am, just 2 minutes. We are having some trouble in the sharing screen. Sorry for the inconvenience. Give us 2 minutes. Good afternoon, everyone. Am I audible, ma'am?
Yes, yes.
Okay.
Continue.
Good afternoon, everyone. My
Good
name
afternoon.
is Kavya Rawal, along with Ashvi Chaudhary. We are both from Department of Microbiology and Biotechnology, Gujarat University. We are currently pursuing MSc Microbiology. Today, I am presenting my internship experience at the Central Laboratory of Gujarat Pollution Control Board, Gandhinagar. Firstly, the introduction about GPCB. The GPCB, commonly known, was established by the government of Gujarat on October 15, 1974. In accordance with the provisions of Water Act 1974, there are its primary mission is to protect the environment, prevent pollution, and control water pollution within the state of Gujarat. The major objectives of GPCB are environmental protection and pollution control. It works in improve environmental quality, monitoring pollution sources, manage hazardous and plastic waste, and ensure industries comply with environmental standards. The Central Laboratory at Gandhinagar performs chemical biological analysis of water, wastewater, air, and industrial samples. It is equipped with advanced instruments such as AAS, HPLC, GC, MS for accurate environmental monitoring. AAS is the atomic absorption spectroscopy, which is used to detect the metal ions present in water. Analysis of water. Water quality analysis is important because water can be contaminated by chemicals, heavy metals, fertilizers, and microorganisms. Various physical, chemical, and biological parameters are tested